Okay, so we're going to talk a little bit here about specification. So we're still kind of in our um, um, early cell stages, right? So we're building this ball of cells, but at some point we're going to start doing cellular differentiation, right? So the early cell stages, they're all kind of just stem cells, right? So they don't have any specific function. They just have undetermined future at this point. So there's got to be some sort of signals that say these cells are going to turn into skin, and these cells are going to turn into muscle, and these cells are going to turn into eyes, and yada yada yada. So we're going to talk a little bit about how that process occurs through what these specification strategies. And so let's turn on the pointer, even though I hardly ever use it because I'm always on the wrong page here. All right, so we have this process here called where we have specification, determination, and then differentiation. So commitment is the process of specification and determination. Specifically, specification is where the fate of the cells or tissue is capable of differentiating autonomously, so autonomous specification, which means by itself, when placed in a neutral environment. So if you take the cells and you put them in a petri dish without anything, they're all by themselves alone, without external factors, I guess, which will make sense shortly here, that they'll differentiate, right? So, however, it can change, they can differentiate by themselves. However, they can still be changed by their neighbors, right? So determination is capable of differentiating autonomously, even when placed in an area of different neighbors. So at this point, so specification is they can differentiate but still have the ability to change. Determination is they are able to differentiate, but they can't, their neighbors can't influence them. And then differentiated is when they are their final product and are no longer able to change. Or they're so the difference between determination and differentiation. Determination means they're not differentiated cells yet, so they're not like muscle cells, but that's all they can ever be. Like they can't switch that. They will become muscle cells, they haven't yet, but their fate is sealed. Specification, at that point, they are not, their fate has not been sealed yet. Okay, so in this example here, we have two different cell types, right? Those are to be muscle, which are the pink ones here, and those that are going to be neurons. And this is from our early blastocyst here. So if muscle cells placed with neuron cells, a change of the neuron or of this muscle cell to neurons would be an example of specification. So you can see here, even though it's a different color, you see this muscle cell, what was supposed to be muscle cell is surrounded by all these yellow neuron cells, and because it's surrounded by all those yellow neuron cells, it itself becomes a neuron cell. It's pink to show that it was originally the muscle cell, but you can see by the shape that's a neuron cell. So that's if the specification is labile, so able to be specified. If it's determined already, you can see that even by placing this muscle cell in the neuron cells here, that the muscle cell still develops into a muscle cell even when surrounded by neurons. So this is determination. This is already determined what it's going to be. So specification versus determination. Specification can change based on neighbors, even though it was predetermined to be a muscle cell. The neighbors can still change it. Specification, irreversible. If it's irreversible, it's determination, right? Okay. So here's some characteristics of autonomous specification. So the fate is determined by cytoplasmic determinants. So this means when we get to conditional specification in the next lecture, that is where your neighbors influence you. So kind of like we showed previously with that, the muscle cell being affected by its neighbors. So an autonomous specification, fate is determined by things like proteins or mRNAs within the cytoplasm of the cell. And this development proceeds in kind of a mosaic pattern. So there's a myriad of cells, each following its predetermined path. So if isolated from the embryo, so if you take one of those cells away, kind of like we did with a muscle cell, the cell will still develop according to its original fate. And this often occurs in early development of organisms like nematodes and tunicates. And it results in invariant cell lineages, while cell fate can be mapped from the earliest divisions. So it's easy to map where in the embryo the heart tissue is going to come from, because the neighbors aren't going to affect it. So it's predetermined very early from the cytoplasmic proteins and mRNAs. So we know what early on what cell is destined to become what type of tissue. So here we have, even if you are like early in this trochoblast here, there we go. If you isolate the cells from the developing embryo, their fate's been determined based on their position. So even if we take it from this early cell stage and isolate it and nurture it, culture it itself, you'll see that you'll still develop this same tissue. It's kind of hard to see under my picture, but you have them in the slides. That you'll still have this same tissue develop, regardless of if it's by itself or in the entire embryo here. And so this arises from these morphogenic factors, proteins, mRNA, that are within the cytoplasm of the cell. So you can see here we have a single cell stage, and there's this developmental biology researcher, his name was Conklin, and you notice that within the cytoplasm we had these different colored parts, right? So we had this kind of segregation of towards the bottom here, this like he called a yellow crescent, or like in this case it looks kind of orange, and a much lighter color at the top. And he noticed that the cells within the yellow cytoplasm developed into muscle tissue for the C-squirt, and that at each stage this yellow stuff was getting divided into whatever kind of the local area is. So in this case, it divided onto the cells at the bottom here, because it was all located in the bottom in the previous cell stage, and that morphogen, likely because it's colored like this, likely a protein, right? Because mRNA you wouldn't be able to see, it would be clear. But anyways, that developed into this muscle tissue because it just so happened that those cells inherited that morphogen, and that's kind of what led to that. So as we said before, if you were to remove these cells at the bottom here and then culture it by itself outside of the cell mass here, you would be growing just muscle tissue, right? And so you can see these or endoderm and muscle tissue. That's what kind of segregates towards the bottom, at least for the C-squirt example. But you can see these different morphogens are in different areas, and those different areas of the egg, or very early fertilized egg, I should say, are what determine what those cells will morph into or develop into. So he found also if he would remove cells from this area right here, B41 layer of tissue, that the C-squirt would develop with no tail or muscle. So we can see that in this next picture here. So if he removed that B4 section, let's go back and look what that was. This was kind of this bottom section right here. If he removed that and then looked at stained and looked at this developing sea squirt, that you can see everything up here at the top, that's all the same because that was at the animal pole of the fertilized egg. However, down towards the bottom here, where the actual B4 was it B4-1 early cells were, that no longer develops. That development is all messed up because all the offspring of that cell, the daughter cells from that B4-1 cell, don't exist now because they've been removed. So that tissue, which all has to arise from that B4-1, will not develop. And so here, this is kind of what we call fate mapping. So you can see that you can develop complex fate maps based on this. So we had B4-1, which was early on in this embryo. And based on these studies and like removing single cells like this, we can see what each early stage cell will eventually develop into. So B4-1 is endoderm, a little bit of mesenchyme, and most of the muscle tissue. So if you remove this B4-1 and let everything else develop, you'll be missing this. And you can see that this A4-2 cell, which if we go back and look 2 slides ago, A4-2 would be behind this one. Or no, sorry, A4-2 is right up here. Or wait, this is lowercase a. So was that? Yeah, lowercase. What one was I looking at here? Yeah, lowercase A4-2 would be top left here, right? Yep. So that one, if you would remove that, you would no longer have brain for this animal. Well, you'd have brain stem, but all that tissue arises from that single one, and there's no changing that based on your neighbors or anything like that. Okay, so even more interesting, so if you separate developing embryo tissue based on their location, A4-2 or B4-2, the cells will develop into the respective tissue regardless of being removed from the embryo. So if we did remove that A4-2 and let it develop, we could just grow just brain. Or in this case here, you see that this B4-2, we would get just ectoderm for both of these tissues. If you were to pull this A4-1 out here, where we'd have endoderm, notochord, and a little bit of nervous system, you would get just that, notochord and endoderm. I guess they don't show a little bit of that nervous system, but here we have muscle and mesenchyme and endoderm. So if we would pull just B4-1 and develop that, that's all we get in our final organism. So the fate is sealed early on. So also interestingly, if you transplant that yellow cytoplasm that we saw at the bottom of that cell from one cell into another, that cell will then also become tail. So remember that yellow cytoplasm, those morphogenic protein factors that were in the cytoplasm, makes muscle and tail of the sea squirt. So if you take that stuff out and put it into a different cell, that cell will also become tail. So those are the causal factors that are making this tissue develop into tail. So within that protein and those morphogenic factors in there, one of the main causal agents is an mRNA called MACHO, M-A-C-H-O, like macho, which encodes a muscle-specific transcription factor. So the cells that form muscle rely on macho RNA being present in order for those genes to be expressed to differentiate and create the proteins that are specific to that tissue. So macho protein, which is stained in dark purple or black here, so down at the very bottom, localizes on the lower side of the cell. So after the cell splits, the mRNA is then localized in this B4-1 section. So it's located specifically, so after we get to this 4-cell stage here, 4 or 8, I can't tell the depth here, that it's only located in this one cell, right? So it's localized that way. And we will talk about how these gradients are created to localized to a single cell like that. So if you take macho and you knock it down, so you get rid of that protein in the mRNA, this results in fewer cells that will differentiate into muscle tissue. So this is knocked down, not knocked out. So it's really hard to, like with CRISPR, you can remove a gene. At the time of this work, we had RNAi, so RNA interference, which is a way to, we say knock down because you get what's called leaky expression. So a few proteins are made, way less, like you turn it down, you knock it down, but you don't knock it completely out. So that's why I say it results in fewer cells differentiating the muscle tissue, because we haven't gotten rid of all of the macho, but we've gotten rid of the most of it. And we can see that the amount of macho is directly proportional to the amount of muscle tissue being created. So if you take macho out and add it to other blastomeres, so other developing cell masses here, then you get more muscle tissue. So like I said, it's if you increase it past what's normal, you get more tissue. If you decrease it, macho, the concentration from what is normal, you get less muscle tissue. So, all right, that is it for autonomous specification. We will get into conditional next, and then syncytial is the last one. So thanks for watching, and I'll catch you in the next one.